Okay, so let's get started by talking about clammy. So the first prompt is, the value of using Chlamydomonas. Is that how you say it? All of a
Yeah.
sudden I felt a little bit,
A bit weird.
yeah, as a model. So
Yes.
what is, like, at a high level, what is this organism? Where would you find it in, a natural ecosystem, and like, what's it doing there?
Yeah. Okay. So these are all great questions. So the organism specifically that we study is called Chlamydomonas reinhardtii. That's the species, and it is a small green alga, and it's a unicellular eukaryote. So it's a single cell, and that is very much similar to yeast. So you can think of it. Sometimes it's often called the green yeast because it also shares a lot of plant-like characteristics. So it has a chloroplast because it undergoes photosynthesis, but it has many characteristics of both plants and animals. And so it's sort of, sometimes it's called a planimal, it's like lives right in between, and it shares a lot of the characteristics. So some of the genes, for example, that it has are very, very plant-like, while many of the structures and the functions of many of the genes are very animal-like. And so it really is a complete hybrid of the two. And that really gives us a lot of benefits and what to study, in addition to the fact that it's, a laboratory sort of powerhouse. So because it's a single cell, that's sort of isolated, you don't have to like separate it from the rest of the tissues and things like that to get it to survive. And it also has a lot of benefits in experimental tractability. So the doubling time is like 6 to 8 hours. So you don't have to wait like days and days for things to grow up. And it's, stable at room temperature. So you don't need like special temperature controlled conditions. And as we go on, we can tell you some of the more other advantages. But basically, it's a green alga that lives in, fresh water. It can live in salt water. It lives in soils. It lives in lakes. And really, one of the really interesting features about this organism is that it is been very highly adapted to a lot of different ecosystems. So it can survive in snow, for example. So different types of species of Chlamydomonas, these algal species can really readily adapt to their environment. And that's actually one of the things that's really exciting to us in sort of the Arcadia sense, is to think about how this organism can adapt to different environments. But it is a really powerful model system because many of the things that we are interested in studying, that I'm interested in studying scientifically, which is really related to the cell biology of this organism. So and particularly the cytoskeleton. And these, when you think about the types of things that, are typically models are used to study, it's really because of the ways in which they are conserved with human biology. And so that's sort of how originally I got started in this area. But of course, the things that I've always really found fascinating about this organisms are not just the ways that it's similar to humans, but the ways in which it's different that can help us understand the fundamentals. But yeah, so this organism is really, like, to get back to your question, it's found often in soils, and all of the laboratory strains that we have were actually isolated out of sort of nutrient-rich soils, from actually largely a single source. But it does do a lot of these really important features in sort of soil stability, and it is, because it's found in these lakes. It's this part of a rich ecosystem in which it is often sort of eaten by other protists, and then it's, of course, again, a photosynthetic organism, so it does carbon fixation, and there's a lot of interest in studying sort of like marine algae for the purposes of investigating sort of the climate aspects of it too. And then finally, it's often very useful in terms of synthetic biology, because it's actually a really widely used platform for expressing exogenous genes, and for biofuel production. So under certain nutrient poor conditions, you can sort of get it to be a really great platform for biofuel synthesis, in addition to expression of a lot of other things for production of different synthetic products. And that has been something that has been really, really more well developed over the recent years, because you can, it has a lot of tools that are available for expression and, for, different types of manipulation of the, I need to turn off my own. I don't know if you can hear. I don't know if you can hear all. Oh, that's good. Okay. I keep hearing all of my things and
notifications.
Yeah, notifications, which it's like, I don't know how anyone can get this many emails in one sentence, but yeah. So basically it's got a lot of important genetic tractability and, benefits for doing synthetic biology. So really it's sort of like a biology and biotechnology powerhouse.
I love that. That's very cool. So what were some of the, what's the origin story of Chlamy as a model? Was it is there it started in one field and then other people sort of started studying it too and realizing this potential powerhouse
I would say that over the sort of like the decade plus that I've been studying it, I've always been really have really understood it as a system that came into more popular usage, really for study of a particular feature that is what I was studying. So basically, I mentioned it's a single-celled organism, and it has these two projections that stick up from the surface that are called cilia or flagella sometimes, and it's the same structure. But basically, these are antennae that stick out from a cell that are found in actually everywhere. They're ubiquitous. That was present in the last eukaryotic common ancestor, but also throughout the tree of life, including almost every cell of the human body. But it's a really sort of cytoskeletal structure that functions as an antenna. And so meaning that it can receive signals, send signals, but it also in many cases can be motile, meaning that it's used for organisms to swim. And so that's what it does actually on this alga, is that it actually has these two and it does the breaststroke. So it basically does it does the breaststroke and sort of swims in this helical pattern when it's in liquid media. But basically the study of these structures, and they're comprised of a cytoskeletal protein called microtubules, and those structures are really what Chlamydomonas, where it really shines. And it's been under intense study for, since the, mid 1900s, where it became really actively used and developed as this model system to study how these cilia function, how they're assembled, and what happens, when they break, which is something that actually in humans causes a lot of different diseases. But we have been always interested in the real fundamental biology of how the structures are built and how they can be regulated, how their length is set, things like that. And so, that has been a sort of a rich area of understanding. But again, the other aspects in which it's been studied is for photosynthesis, actually. For photosynthesis, it's so largely if you go to a Chlamydomonas conference, it's always often split up into those two, where you have people who study the cilia aspects and the people who study the photosynthesis. And then over time, there was a period of time where biofuels became very, like a sort of hot area of study for, and then now it is very much like synthetic biology is as more tools become available, it's becoming more and more studied for that purpose.
So when you're studying it for its role, to study these breaststroking flagella cilia, do you grow it under photosynthetic conditions, or are you growing it off of like a rich media or something like that?
Yeah, so that's a great question. So basically, so these cells can use nutrients, or they can use light, and so we're actually having it in both conditions. Let that they grow very rapidly under those conditions, and so we grow cultures under conditions where they have the nutrients that they need, but they also are in constant light. And so if you wanted to, like, for example, synchronize the cells, so in the cell cycle, you could have like them cycling in light and dark, but we sort of have them in constant light. It helps them grow faster.
Cool.
Yeah.
Okay. So I think that kind of covers at a high level, like coolness of Chlamy. So let's get into, the value of studying Chlamy as a model for the cytoskeleton. You kind of hinted at a few things, set us up really nicely for that. But I would love to hear like your personal story of how you became a Chlamy person.
Oh, it's so great. I love the story. Thank you for giving me the opportunity to tell you how much I love the system still to this day, and how excited I am about, the new approaches that we plan to take at Arcadia too. So basically, I had, as a graduate student, I was working in a lab also studying cilia, but I was studying it in mammalian photoreceptors. So you may or may not know that the eyes, that you used to sense light in all these organisms, the light is sensed by photoreceptors, which are cells that are neurons, but what is lesser known is that the segment of the photoreceptor cells that sense light are actually highly modified cilia. It's like the same structure.
Is that the rods and cones?
Yes, the rods and cones, the outer portion of the rods and cones that actually capture light, those are cilia.
I did not know that.
And actually, they're highly modified in that they have a very different membrane structure, but the core, components, assembly mechanisms, proteins are the same ones that are found in these algae. Okay. And so as a graduate student, I was studying photoreceptors. And as you can imagine, there has been many, many decades of study of like how we sense light and how visual transduction happens, which means, capturing of photons and turning that into an electrical signal that signals to the brain what we're actually seeing, right?
Yeah.
And so that process has been studied for a long time, like understanding how you get, changes in proteins that sense photons and how that starts a signaling cascade that results in, hyperpolarization or depolarization of a cell, and so that causes the transduction of that signal. So that means that at the time that I was studying it, people sort of thought of that as a solved problem, right? Like almost like every single gene that was involved in phototransduction had been knocked out in mice. We knew what all of them did. We knew what like all of the diseases those things were associated with. But at that time, it was just becoming known that this structure is also a cilium. And we knew very little about cilia and ciliary trafficking and, what motors transport what things. And it's actually a really interesting problem in photoreceptors, how things traffic, because these outer segments that are actually the ciliary portions, those things get turned over completely every 10 days. So when as you are seeing with your eyes, the outer segments of the photoreceptors, those membranes get completely turned over in their entire length and get eaten up by the neighboring, like sort of housekeeping cells completely every 10 days, right? So that means that all the proteins that you need to see, they get turned over that rapidly. That means they need to get synthesized and trafficked to where they need to go continuously. It's like a huge trafficking demand, right? But we didn't understand how any of that trafficking worked. And if you looked at all the other systems in which people were studying cilia, they knew cilia was, really important sort of structure in which you have this dynamic trafficking of signaling proteins to do this sensation thing. But they didn't know like, what are those cargoes? What are the motors? What's happening there? But in photoreceptors, we had an advantage because we knew everything about phototransduction. We knew every gene that had to be, every protein that had to be trafficked there, what it did, what happened when it didn't do what it was supposed to do, or if it didn't get where it was supposed to go. We knew everything about that. And then after the fact, we learned that this is really also a cilium. So now we knew what all those cargoes were. And so we were at this like unfair advantage in studying ciliary trafficking in this system where all these other things were already known for different reasons, for understanding how light and visual sensation worked, right? So then, it turned out that in trying to, combine these two sort of fields, every single paper I read about cilia and ciliary trafficking, I could trace all of the sort of literature back to green alga, like this weird single-celled organism. Everything we knew about cilia sort of seemed to come from this literature. And then when I was done with my graduate school and it became time to decide, well, what do I do next? It just seemed like such a powerful system to understand mechanisms, work in something that was like faster than working with mice, and just was something that was, like we had really great genetics because there were all these like, it was great for forward genetics, meaning that you could like, randomly mutagenize an entire population of cells, do a phenotypic screen, and then, identify novel functions of the genes that were mutated. And then, so there was a great library of genetic mutants that have been identified in this way. So there was so much potential for understanding this problem using a really powerful genetic system. And then it had all these other advantages, like you could actually separate the cilia from the rest of the cell really, really easily because the cells themselves, if you put them in a sort of unhappy condition or put them in low pH or something, they just pop their cilia right off. The Chlamydomonas cells do, like very cleanly right at the base. And so, and then you can obviously purify the cilia, do proteomics, and then you can do all sorts of different kinds of analyses this way. And that's really hard to do in any other system that has cilia. This is like unique, even in human cells. It's like all of these cells you could technically biochemically purify the cilia away from the cell, but it's really hard. There's a lot of contamination and, cross-contamination of different components. But in our cells, they just do it automatically. and it's very clean right from the base. So there's all these
Mhm.
fantastic advantages. And so when it was time to do a postdoc, I thought, oh man, like I cannot imagine doing this in any other, continuing this work in any system other than this. And I even did. I went and interviewed at places that studied sort of cilia from all these different model organisms. And I like fell in love with this one. And I still to this day, and maybe you can tell my enthusiasm.
It shows.
It is not, after all these years, it's not worn off. And and that is only actually, so that's only half of the story, because that tells you about the part that is conserved with humans. And that doesn't really even come close to tackling why I am so excited about this system today, which is that throughout the course of my postdoc work and from my own independent lab, we discovered that, the cilium that is really microtubule based, so the larger cytoskeletal components, we discovered that there was a huge role for ciliary assembly and trafficking through the actin cytoskeleton, which is the the smaller component, like the thinner component of a more flexible component of the cytoskeleton. And at the time that we discovered this, was such an uphill battle convincing anyone that this was true, because everyone thought, like, what does actin have to do with cilia? This is a microtubule-based system. Everything about it is microtubule-based. Trafficking to the cilia is microtubule-based. Trafficking in the cilia is microtubule-based. Everything about it is just microtubules. And if it's actin, that's how you know it's not related. And we found that, of course, people who study actin are probably not surprised because actin does everything. But we had discovered this. And the really spectacular thing about Chlamydomonas for understanding this problem is that, unlike virtually like all of these other, organisms, Chlamydomonas has two different actin genes, one that looks exactly like humans and one that is as different as actin can be. And that in the same cell. Okay. And that is like absolutely mind blowing, because a lot of cells actually do have different actin isoforms, but often they're all almost exactly the same. Like humans, for example, have 6, but they're almost identical. They're just only a couple of, amino acids different, right? But in these cells, there's one that looks just like the human version. So we can use all the tools that we use to study the human version. And then there's this other divergent actin that is so different that it's as different as it can be without it not calling it an actin anymore. Pretty much.
Yeah.
Right. And but now you've got a cell where, the ways in which actin is regulated, you can think about like just the cytoskeleton in general. The reason why it's so important and interesting is because you basically have like, either a single protein or a dimer or something, and then it can turn from that into a polymer that can do all sorts of important things in a cell, right? It can traffic, regulate membrane dynamics. It can, it can do any number of different functions within a cell, but it's really the combination and the way in which that happens is largely from going from a monomer or a dimer into a polymer, into these filaments that can do work, that they can be bundled together. Motors can traffic on them. They can, form shapes that can deform membranes. They can do all sorts of different types of work. And all of that happens through dynamic regulation of like a single protein or two proteins, right? And that is like, just first of all, just unbelievably cool, right?
But the complexity that arises from
just that flux of like monomer to polymer, right? Or dimer to polymer. But then that means like, how does it know if it does 100 different things in the cell? How does one, polymer that does so many different kinds of things, how does that happen, right? And the answer to that question is really that it uses regulatory proteins, right? So it has different proteins that function on this polymer in different ways, that are localized in different places, expressed at different times, things like that. And that gives rise to this like great amount of complexity. But in Chlamydomonas, now you have an actin that's very, conserved and an actin that's very divergent in the same cell. And you've got then some amount of regulatory proteins in there that dictate, first of all, do these two proteins can they copolymerize? Are they totally separate, filament systems? How does a regulatory protein know which one of these things to go to? Our sort of naive conception was that these regulatory proteins, there might be some that interact with one actin and some that interact with another. All of our work has shown that these proteins can interact with either one of them, and that they are very highly redundant, even though their sequence is like wildly different. So everything we understand about how actin works and is regulated is wrong. This is, and the only reason we know that, is because we've got a very, very different actin in the same cell. So if you go to the way science is normally done, let's find the, systems that are most similar to humans,
Sure.
right?
Yeah.
Now we've got a case in which we've got something that is so different from humans that really it's hard to get anyone excited about, because they say, this is like a weird actin and a weird cell. Where else is it even found? It's like this really special case. But just from what I've told you today, we can say that our understanding about how regulatory proteins work, how promiscuous they are, how much they're able to interact with things, with very different sequences, our complete understanding of how these proteins work is upside down as a result of looking at the thing that's different, right?
Right.
So that is like the real power of understanding how when biology has solved a lot of different interesting problems, it gives us this deep insight into the fundamentals of how we understand, in this case, cell biology to work. And it's only by looking at what's different, not what's conserved. And that is very much the promise of Arcadia.
Right. Love that plug. So close the circle for me on the story of what actin's doing in the cilia of Chlamydomonas. What was that story?
Yeah. So it turns out that like is always the case in many different systems, actin is doing 100 different things, and it is actually doing so. And so we found roles for actin in regulating, expression of ciliary genes. We've found roles for actin in trafficking of membrane proteins from the Golgi. So when you get synthesis of membrane proteins, and then it has to get trafficked from the Golgi to the base of the cilium. So the Golgi in our cells is very close to the nucleus, and the cilium is like at the apical part of the cell. So it's a long distance for it to go. All of our understanding of that trafficking we thought was microtubule based. And that is still true. That is very strong evidence that there are some proteins that are being trafficked on microtubules. But we found that there's actually an absolutely critical role for actin in trafficking from the Golgi. And that could be either short range transport from actin from the Golgi to microtubules, or all the way long range transport all the way to the cilium. We've also recently found, roles for actin in regulating membrane flux that's already been trafficked. So not just from the Golgi, but from the plasma membrane through endocytosis. We found roles for actin in dictating what goes in and what stays out of the cilium. So there's a gate, there's a ciliary gate that dictates what goes in and what stays out. And that is very actin dependent. So through this model system, we've actually found many different, we've been able to dissect all of these different functions. And that is largely because of Chlamydomonas. Because if you, for example, were to do this in a mammalian cell, where you have basically one primary cytoplasmic actin that if you start to perturb it, the cell basically dies. But because we have a second divergent actin that is not susceptible to the same, type of, inhibitors and things like that, because it's so different, we can manipulate the actin in the cell and still not kill it, and dissect all of these different roles. So again, once again, what's different about this cell allows us to study, actin dependent functions in this unique way and dissect all of the different properties.
That's really cool. So have people been able to validate, in human cells or in other more animal cells, that this actin microtubule situation happens in other organisms other than clammy?
Yeah. So it actually turns out that there's a lot of evidence now. When we first started this work, no one believed it. And then it sort of started an explosion. There was an explosion of research in this area. And now, human cells have different properties. So for example, Chlamydomonas has a cell wall. And so typically actin is used to be part of set the shape of a cell or their morphology of the cell. And so because we have a cell wall, we don't need the actin to make that thick cortical, dense network. And so cells that, for example, in human cells, they don't have a cell wall. And so they actually need a dense actin cytoskeleton to do that. So some of the differences that we see are as a result of the, basic morphology and shape of a cell relative to ours. But those other properties, it's absolutely the case that the membrane dynamics and things like that, many of the sort of, roles for actin that we've uncovered are really, really well conserved functions. And the one that we have just recently found, this role in endocytosis, we now have new evidence that we think, that we can support that using, different types of manipulations that we know to also work in all sorts of cells, including in animal cells. So I think many of these things have panned out, but again, we're still just as excited about uncovering sort of what's unique about this biology, because it can help us understand the fundamental properties of cytoskeletal remodeling and trafficking and all of these things that you wouldn't otherwise study if you only studied everything that was, the same, right? Because, and it also helps us think about how we might engineer different systems, because when you see how different organisms have engineered, the ability to manipulate their cytoskeleton in certain ways, that's something that can, of course, be useful, to do or introduce or manipulate in human cells as well, not in the ways that it's already being done, but in a way that we might co-opt from other systems.
Could you give me 5, just like, I don't know, 2 minutes, 5 minutes, something short on this endocytosis story, because I'm personally curious.
Oh yeah, absolutely. So this was like just so remarkable. And I'll give you like a basic rundown of the thing that really surprised me about this. So basically Chlamydomonas has, these cilia that are sort of, full length, which is about 10 to 12 microns in a healthy cell. And like I mentioned to you before, you can actually induce them to pop off their cilia. And a really nice feature of the fact that they pop off is that you can do that across the population. So you get 100% synchronization across this. So in a way that you could never do for the cell cycle, right? So you can pop off the cilia from 100% of the cells. And when you do that, they start to regrow synchronously. So that means that you can even like measure how they grow and the rate and all of those things. But the interesting thing that we've known for decades from work done, many, many years ago from people who came before us in this field, is that if you pop these off and then you were to, for example, treat the cell with like a protein synthesis inhibitor that doesn't allow any new ciliary proteins or anything to be made, these cilia still grow to half of their normal length. So that means that there's already a bunch of ciliary proteins like hanging out in the cell, right? So we already know that to be the case. And really virtually no matter what perturbation that we do to a cell, if you sort of deciliate the cell and let it regrow, even if you screw up some sort of mechanism that in making of ciliary proteins or trafficking of ciliary proteins, you still basically get growth to half length because it already has all that protein available. It just needs to like assemble, right? So, in trying to understand like how can we dissect the roles of actin in ciliary assembly, we were looking at different actin regulators. One of them was for a branched actin regulator called the ARP23 complex. Basically, it's a complex of proteins that allows for actin branching. And that's one of the things that's involved in endocytosis and also like leading edge migration of cells that can crawl, things like that. And so we had a mutant in one of the proteins in this complex, and it didn't grow cilia at all. Like after severing normally almost everything, it's almost impossible to get a situation where you don't at least get to half length. You couldn't get any growth at all. And that was really surprising. Like, that is weird. Like I've never seen this actually happen in myself, and it turns out that what that means really, and that also happened in the case where we had used inhibitors to block that same protein complex, because you could argue that maybe there isn't like an excess pool of protein in the cells that, in the mutant cells. But we also found the same thing with inhibitors of the same protein. They also couldn't grow cilia at all. So where we knew there was excess protein. So it's not protein, then what is it? It's probably membrane. Because you also, in the cilia themselves, they're a microtubule structure, but they're ensheathed by a plasma membrane that's the same, that's continuous with the rest of the cell. So you need basically microtubules and you need membrane in order to grow cilium, right? And so if you have enough protein, we already know there's enough protein, but it still doesn't grow, maybe it's membrane, right? And then we also did some more experiments to sort of exclude the fact that it was like Golgi-derived membrane, because even that very early growth, the one that happens like long before any Golgi protein could ever get there, that was not happening. So we thought, well, the membrane's got to be coming from somewhere else, right? And then, of course, like from other work done by other people that have been studying actin for a long time, in yeast, for example, we knew that this complex of proteins that regulates branched actin formation is involved in endocytosis, right? So there in yeast, for example, there's endocytic patches that are filled with branched actin that are shaped by branched actin. And so we thought, well, maybe we have the same sort of thing going on where you have, endocytosis mediated. And this is how little we knew about this system. Like no one had ever characterized endocytosis in Chlamy. Before we started doing
yeah.
Like it's hard to remember, like when you're working in a weird system, like how much, people have only worked on what they've stumbled across.
Right, right.
It's like comprehensively studied, right? No one had characterized endocytosis at all, or even knew if it was happening in Chlamydomonas. So we thought, okay, well, we need to figure this out because that membrane is coming from somewhere. It's not coming from the Golgi. Where is it coming from? And we know that the ARP-G3 complex, this protein complex we're studying, is involved in endocytosis in other organisms. So we started to characterize that, found out that there was in fact like evidence for endocytosis, not just of, of membrane, but all, where we could see dye internalization was impaired by these mutants. And we could also see proteins that we knew were destined for the cilium that were in the cell membrane. The endocytosis of those proteins was not happening. So we basically showed that, that by blocking this complex that we know is involved in endocytosis, we had an impairment in, reclaiming of ciliary proteins that were destined from the membrane, and we couldn't get any sort of ciliary growth. So we basically proposed a totally new model for ciliary trafficking, where for early proteins, when you sort of really need to grow a cilium fast, right? But you can't just wait for proteins to be synthesized and go through trafficking pathways and get traffic long range to the base of the cilium. When you really need to grow immediately after severing, that you need to incorporate membrane much, much more quickly than you can get from elsewhere. So basically, we proposed a new mechanism by which you get, rather than long range trafficking or even like lateral transport, that you basically have an endocytosis dependent mechanism in which you reclaim membrane and ciliary proteins and then route it to the cilium when you need to do it quickly.
So it's kind of like all of that, those components are kind of sitting on the membrane.
A reservoir.
Primed.
Like a reservoir.
Yeah. And then they get sucked in and then shot back out.
Yes, exactly. Yeah, exactly.
Really
so basically it's like this plasma membrane is serving as a reservoir for proteins and membrane for when it needs to happen quickly. And then if you can wait long enough, then you can get all these other sources where you can get new protein synthesis, new trafficking from the Golgi by the conventional mechanisms. But if you need it to happen fast, it's already ready. There's a reservoir in the membrane for previously trafficked proteins. And then that process is actually not just, you could imagine theoretically that it could be like laterally transported, but we actually showed that it is an endocytosis dependent mechanism.
Very cool.
Cool, right?
I mean, awesome. Okay. Okay. Excellent. So that was prompt 2, value of studying cytoskeleton and cilia and clammy. So let's talk about prompt 3, which is the major open questions in the field. So this is, and this can be your personal research. This could also be, just like wide open questions that you like are like, clammy would be perfect to study this or that.
So for me, I think there's so many fundamental like cell biological questions remain unanswered, and it's really easy, especially when you study something as taught as the cytoskeleton, right? Like under every undergraduate learns about, the cytoskeleton and microtubules and actin. If you're studying biology, often you learn about these things. And it can feel like this is something that is everybody knows everything about, but the field is still so young. Like almost literally it's only been like 20 years since we've known any molecular detail about, the actin cytoskeleton, for example. So many things remain unanswered, even understanding what all of the actin binding proteins are and what they do. Like so little is really understood about that, right? And understanding this fundamental question, for me, I think that this is a really big question, is trying to figure out, how that thing that I mentioned earlier is, how is it that you get such functional diversity from a single protein, right? Like you have like immense functional diversity, and that's from the combinatorial power of its interaction with a huge host of regulatory proteins, right? But although actin itself is one of the most conserved proteins on Earth, its regulatory proteins are extremely divergent. But why? Right? And that tells me something. That tells me that we haven't even scratched the surface of what actin is capable of doing, right? We've only looked at the ways in which actin functions in the way that conserved regulatory proteins are functioning. But the fact that there are so many divergent regulatory proteins, right? Just even across organisms where the actin itself is the same, the regulatory proteins are wildly different. And so that means that there is other functions on the table. There are other, and there are other interaction, that are possible. There are other, that the possibilities for engineering the system are endless, right? Like we just haven't even scratched the surface because we have only been studying what's conserved. And that's what I've been like truly excited about, about being able to sort of like, with abandon, go and pursue the kinds of questions that I think will completely transform our understanding of what the actin cytoskeleton is, what it does, and what it's capable of, right? So that is like truly, it's just such a gigantic question that we can really only understand by looking at the, diversity of life and doing comparative biology to say, how are these regulatory proteins different from these others and these other related species? And how are the functions of the actin cytoskeleton different in these two? To do these sort of genotype-phenotype correlations and really understand how these can function, both now looking in sort of situ as a cell biologist would by labeling things and looking at them in the microscope, but also by doing the biochemistry and studying how these properties of, actin polymerization and dynamics can differ, by studying it in vitro, right? So that is also like really, really powerful. So I have been like very, very excited about doing, studying the actin cytoskeleton for this reason. That's a really big, important question. But even the things that I told you about before, intracellular trafficking. And, for example, we have Chlamydomonas that has this microtubule-based network where and now we've established a new actin-based network through which all of this trafficking can happen. But these are still only, cilia that are at the apical surface of the cell. And like they are often in human cells, they are often just the apical surface of cells. But then you have other organisms like Tetrahymena and protists that have hundreds of cilia all over their surface.
Right.
Like really dramatically different polarity, right? So it's, now you've got cilia and you've got to regulate how things get trafficked there, how the length of the different cilia in different places can occur. And so now that is a totally different question, right? Like we have the ability to, these cells have the ability to resolve their trafficking in a way that no one has really studied. Right. Yeah. So again, all of these, fundamental questions that we think we understand, right? We think we understand how trafficking works. We think we understand how, cytoskeletal polymer regulation works, but only because we study it in such a narrow context. So hopefully I'm conveying to you that even the like basic questions that we think we understand, these are the really big questions because we don't actually know what we think we know. And because we haven't really studied studied how it works through, a diversity of life.
Yeah. It kind of reminds me of like how you reminds me of like my PhD work, which is like or even like in a postdoc, like you study one question super, super, super deep and you don't think about the rest of biology. I know. And it's like the same way in like this field is just focused on like, photoreceptors so deeply and like knows everything about it. But like, what about the photoreceptors in other like kinds of eyes or like, or, like deep sea eyes or,
there's so
like
much
that.
on the table, it's the same way. Sometimes I make the analogy to computational biology where often people think like, oh, if you aren't sort of modeling things to just decide, like what is within the rules of physics, where you say like, oh, I could do 100 different experiments, but if some of those things actually point to a model in which like literally it breaks the laws of physics for that to happen. Like I'm wasting my time doing those experiments by not doing a little bit of legwork ahead of time. And I think we are doing the same sort of thing by not using the sort of the diversity of biology to sort of see what it's already discovered, right? And that means we're wasting a lot of effort in doing experiments that biology already knows the answer to, right? And so that is something that we can really do. And also, it's really unbelievable, like how much, information you can get by thinking more broadly and not just deeply. So I would say that even in the small realm of biology that we're talking about, the fact that we've been actually studying both microtubule dynamics and actin dynamics, studying the cilium and the ways in which actin regulates intracellular processes, including, ciliary assembly. by studying both actin and microtubules, we have made so many important insights because we don't just study the one, because we're like, wait a second, we know that the other, because cytoskeletal polymers actually have a lot in common, even though that they are very different, their properties are different, but the ways in which they're regulated and how they function are very similar. In fact, like when I teach cell biology, I actually talk about all the ways in which they are similar. And so every time we discover something new, we start to think about the other cytoskeletal system in a different way, right? And I think the same type of potential is true is when you start to actually look across the tree of life and even look at related species that have very similar but different genes, but then slightly different functions. Like we can start to understand how these, what the actual do more functional annotation of genes, understand how this biology works. Like our fundamental understanding of most of these things is wrong. And it's like, it's amazing. It's not just wrong, it's incomplete, right? Like, And like, so that I think that is, but it's incomplete, like so severely that we really don't, we are missing the bigger picture completely by by leaving this type of experimentation on the table.
That's just an excellent point. Okay. So let's move to prompt 4, tools. Arcadia is really interested in tool development. What can we build that helps unlock these model organisms for more people to study, more people to think broadly about a wide diversity of life. So you mentioned that Chlamy has a lot of tools, a lot of resources. What, if you could just magic finger, get a new tool to unlock something, what would that look like?
Yeah. So 2 things. One would be getting CRISPR to work really reliably to actually targeted gene deletion. We have we've come up with, the field is amazing. They have come up with so many clever ways around this problem. And they've even there's a couple papers out there trying to get reliable CRISPR to knock out the specific genes of interest. And it's variable efficacy, it's one of these things, anything new, we know it's going to work. It's just a matter of having some concerted effort, time, resources to put towards a problem. That's often the thing that is the barrier for many different organismal communities is that it just needs concerted effort in a way that it's difficult to put together with sort of revolving door of people and not stable funding and all of these things. That's just difficult for, so it requires critical mass in a field and people.
And it's not hypothesis driven. So it's not friendly for writing a grant for.
Exactly. Right. And so if people manage to do it and they inch along fears over the course of decades, they're able to make progress in a field that until it's sort of mature enough where more people are interested in doing it. Or in our case, we actually had to develop all of the sort of methods and tools ourselves to visualize the actin cytoskeleton, because it turned out that in our system, you couldn't even see it. That was one of the reasons. So 2 things actually put the nail in the coffin for us is that, there was an actin mutant available in our organism. It had almost no phenotype. Why did it have no phenotype? It's because there was a second actin gene that was very different. Right. And so everyone just thought if you get rid of actin, nothing happens. So it must not be that important. And then on top of that, the way in which people typically study actin is by visualizing it with the same fluorescent probe. And that didn't work in Chlamydomonas. So the combination of those 2 things, no phenotype in a genetic mutant and the inability to visualize it, everyone just said, oh, well, this is just a microtubule based system. Actin's not really doing it. So but we had so much functional data to suggest otherwise. We actually had to develop the tools to be able to see it. So we published all of the work on actually visualizing actin filaments in Chlamydomonas for the first time ever. We had to sort of overcome that barrier ourselves to make progress and to understand what we actually wanted to do. And that's often what people do in these fields is to sort of invent the technology they need to solve the problems that they have. And that is how these fields move forward. So CRISPR is just one example, but then for all organismal communities, of course, as cell biologists, it's ways to visualize, things in cells. The better it is signal to noise, the better it works for us, the better sensitivity it is, the better it works for us, the better is we're able to visualize it in real time and in time-lapse microscopy, the better it is. So all of these things are things that are continually improving over fields. So when I say that we have some, a lot of different tools. These are things that have been evolving. Like for the longest time, it was really difficult to get exogenous genes expressed in Chlamydomonas because there was a lot of epigenetic silencing of anything exogenous. So then all sorts of things, methods were developed to sort of improve on that. And so over the course of decades, this has actually become a well-supported system. But again, people ask us all the time, what organisms are you studying at Arcadia? And it's and the thing I like to convey to people is it's not about organism because, for example, Chlamydomonas is an excellent model system for studying cilia because the cilia are very well conserved with humans. It has tons of tools to be able to do that. But it's actually a really great non-model for studying the actin cytoskeleton because it's what's different about this divergent actin that's really going to help us understand how the actin cytoskeleton works, not what's similar with humans, but what humans don't have at all.
Right.
That's going to change our thinking about how actin is regulated. So even though this is all one organism, it's not just an organism is not a model or a non-model. Like, yes, there are certain tools that are available to us because it's been developed for a different purpose, but there are still tools that we don't have. And there are questions that we want to answer that have nothing to do with humans. I mean, it has everything to do with our understanding of basic biology, but not it by using, the differences in the system rather than what's the same.
Right. I love that description because it really highlights how when you do get these like tools specific for your organism, or they can really unlock like a whole new learning, a whole new way of looking at things.
Yeah. And if you let me make a 1 second plug, and I actually hope that this is an aspiration for us at Arcadia is to really, that many people are unable to actually, overcome that barrier for very good reasons, right? Because it does require that concerted effort and energy. And often it's just coupled to the problem that they need to solve. But I think we as Arcadia can, for a broad range of organismal communities, we can solve a lot of those problems. I hope we can send people into the future 10 to 20 years and change what's possible so people can ask the right question in the right system and not just be told, hey, why don't you just do this in mouse or in yeast, because that's what has the tools. Hopefully we can sort of solve some of these problems on a larger scale and really make it so that people can start asking the right questions in the right systems and completely unlock, as we've been saying, all the biology that has not been available to us to this point.